This short video is covering NETosis induction in isolated neutrophils. The first slide here is just a blood tube, and we've just let it stand and we've added PMA to it. And what you can see is this sort of white cloud sitting on top of the blood here. And so even with the naked eye, you can see this nondescript cellular material at the top. This is NETs being released after stimulation with PMA. PMA is a really useful molecule to stimulate neutrophils, but it's not a naturally occurring molecule. Building on this principle of stimulating neutrophils with PMA, we developed a model in our laboratory to stimulate isolated neutrophils. And these graphs are extremely important to volition. What they are showing is they are showing a very strong correlation between neutrophil stimulation, neutrophil extracellular trap release, and H3.1. You can see these green triangles that increase. These are a cytox green signal. We talked about cytox green when we mentioned the MRSA in the mouse lungs. So it's a clear sign of DNA release, and it correlates extremely well with H3.1. H3.1 is a key component of neutrophil extracellular traps. If you go back to one of the first talks where we talked about NETosis, and you have the S curve of neutrophil release over time, you can see how these time points align. So at this flat part of the curve between 0 and 2 hours, that's where you've got the nuclear decondensation taking and all those changes in the plasma membrane before the DNA is ejected and released from the cell. And so really we're doing a couple of things. I like to think of it before 2 hours and after 2 hours. All the clinical work we're doing with the research and development teams, and actually further work with the innovation lab as well, is after 2 hours. What do the NETs do? How do they stimulate the immune response? Do they lead to an excessive immune response? What do high NET levels mean in patients? Are there patients with high NET levels and low NET levels, and what does that mean? That's our sort of clinical research after 2 hours. And then the innovation lab are really delving into this period beforehand, this 0 to 2.5 hours. What's happening there? How is the chromatin unwinding? Can we detect patterns of chromatin unwinding? Are there different patterns of chromatin unwinding depending on what trigger you're using? So if we use an artificial trigger, PMA, is it different to a natural trigger? A bug or a protein or some small molecule released by a bug to stimulate NETosis. I think that's really exciting and really interesting research. A key question for us is, can we identify, tease out these different subtypes of NETosis, suicidal, vital, mitochondrial NETosis, and that might ultimately lead us to having precision therapies or discrete therapies for those different types of NETosis. Again, really chasing this goal of precision medicine. What I'd like to do now is take you through one of the first papers published by the Innovation Lab. It's just been accepted in the Journal of Thrombosis and Hemostasis, and we're really grateful to Kieran and Terry and the team for all the work they've put into this. This is just panel A. It's just showing, it's really covering ground we've covered before. It's an EDTA tube, and we're stimulating NETosis. Panel B is picking up a dose response curve. We're just using different concentrations, different strengths of this molecule PMA to stimulate neutrophil release. And you can see when you have the highest dose, 100 nanomoles of PMA, you're getting the biggest response, but you're seeing just a dose response there. In this slide, we're using a NET antagonist, ABAH, and you can see you have a reduction in the strength of the signal from stimulation with PMA. So actually, looking at panel C, what's going on here? We've stimulated the cells with the maximum dose of PMA, and then we're trying to block net release using increasing concentrations of this molecule, ABAH. And as you can see, the higher concentration of ABAH, the more you can block the stimulus from PMA. So you can manipulate, you can attenuate, you can reduce net release. What panel D is picking up is staining with cytox green, and it's really just a mechanism, a way we're showing net release here. What did this part of the paper show? Showed that we can stimulate net release in a reliable, consistent, coherent fashion. And then the very first part of the talk shows those graphs. It's where net release is directly proportional to H3.1. It's where it's proving that H3.1 comes from neutrophils, which is so important. Next, we're going to cover high throughput screening of factors to induce NETosis. And again, this is taken from Kieran or the Zukas paper published in the Journal of Thrombosis and Hemostasis. The problem with the last method I was talking about is it takes a long time to prepare the cells. It's an artificial method, and it's just very laborious and time consuming. So we've used this amazing device. You can see the image taken from BioRender here. It's called a Mantis, and we're dealing with a 384 well plate. So you can test 384 individual wells at the same time. It's quite amazingly high throughput what we can do. The order in which reagents are added is mapped out using the mathematical program MATLAB. And so we can program and test a large number of molecules all at the same time and try and pick out their signal. This actual panel is the control work. And I tip my cap to the innovation lab here, to all the work that was put in to standardize this process, make sure it was controlled and make sure it was really reproducible. And so in the first part, we're just showing again concentrations of PMA leading net release, but it was reproducible and they could do it in a coherent, cohesive, reproducible, consistent fashion. Now, what did we do next? Well, instead of using PMA as a factor to trigger neutrophil release or net release from neutrophils, we used a whole host of naturally occurring molecules to see if we could trigger net release and identify different patterns of net release, which we did. So panel A here is identifying individual factors, which we put together in pools, and then we stimulated and ran them on the machine. Panel E is a list of all the factors that were involved, from tumor necrosis factor alpha, LT alpha, interferon gamma, GM-CSF, granulocyte macrophage colony stimulating factor, all the way down to some interleukins, interleukin 1 beta, interleukin 6, interleukin 18, and some selectins. And actually, panel B is critical to report here. The black dot is just a control. We're stimulating with PMA and proving that activation of neutrophils worked. But what you can see here is 4 different lines, 4 different patterns of net release. We don't know the answer yet, but it's an area we're really focusing on with the Innovation Lab. So we're seeing 4 different kinetic patterns of net release. The question is this typical of, or is this indicative of, those different types of net release that I've mentioned to you before, suicidal, vital, or mitochondrial? Or is it actually just due to a different type or different way of stimulating the cell? But it's starting to give us a window into the different types of net release, and actually showing that net release is not a static phenomenon, it's a more dynamic phenomenon and more complicated than we initially thought. This is a key slide for us because it's allowing us, or identifying targets for us to chase and to pursue further. So when we put all these factors together, we can actually see that there were different patterns of net release from men and women. We've joked about this internally, if I'm honest, is there a difference between man flu and is this a potential answer? I don't think so, but what it does show is that there is differences between men and ladies in terms of their net release. It's possible as we scale this up that we might see this phenomenon more. We do know that autoimmune diseases are more prevalent in ladies, and just showing that there are subtle differences between the immune system in sexes, and that appears to go down to neutrophil extracellular trap release too. And so it's something we'll look at and pick up going forward. This slide here is again picking out the different patterns or different kinetic patterns of net release. And it's really picking out what factors had the greatest impact on net release. So what you can see is tumor necrosis factor alpha, tumor necrosis factor beta, interferon gamma, ferritin and C5a in particular had a really quite marked impact on neutrophil extracellular trap release. It's really interesting because we need to understand and we need to delve into that biology more. Drugs, we have used TNF alpha blockers historically in patients with sepsis. It worked in smaller studies, it didn't work in larger studies. And so are we starting to identify subtypes of sepsis, different types of sepsis where drugs might work? Perhaps there are types of sepsis where neutrophil extracellular traps are more important and types where it's less important. And again, it's really chasing this target of precision medicine, trying to understand why two patients present, perhaps with the same disease, two patients presented with COVID and one would have a very different path to another. And can we enable clinicians, can we give clinicians a tool to start to understand their patient's immune response? And if you could start to understand their immune response in real time, could you change the order, the kinetics in which you deliver therapy and treatments? You might select some treatments for some, not for others. So we're really hoping to use this, or hoping that our research and studies can lead towards precision medicine. And so it's really exciting that we're seeing different types of NETosis and different factors stimulating them. This slide is in again. Again, you've seen this slide before. It's picking up the different types of NETosis that are described, suicidal, vital, and mitochondrial NETosis. And we're definitely not there yet, but you can see a direction of travel for us to try and understand NETosis, try and tease out the different types of NETosis, and see if we can help ultimately bring that to the bedside to help clinicians.